Hi, my name is Camille McCall. I'm a postdoctoral research associate at Rice University working on the Houston Wastewater Surveillance Monitoring Program. This is the first module in this series on quantification of microbial targets. For this module, I'm going to be discussing different methods for RNA and DNA quantification of microbial targets in wastewater. There are three main learning objectives for this module. These include learning the basic laboratory steps involved in quantifying microbial targets in wastewater for wastewater-based epidemiology, becoming familiar with different methods that can be applied for the concentration, extraction, and quantification stages of the laboratory workflow, along with some things to consider when choosing a method. And lastly, I'm going to show a video which will help you gain an understanding of the sample processing methods specifically for SARS-CoV-2 used in the Houston WBE system. Although various methods can be used for sample processing, the following is a standard workflow implemented in our lab and others when determining concentrations of microbial targets in wastewater. This workflow includes measures for proper handling of wastewater samples, concentration, nucleic acid extraction, and quantification. In the next few slides, I'm going to briefly discuss the purpose of each stage, different methods that are commonly used to carry out each step, and things to consider when choosing a method. Here, sample handling includes procedures used to handle the receiving, tracking, and storage of wastewater samples upon arrival to a laboratory facility. Implementing a system to receive and track samples is important to ensure all samples are accounted for and the proper information is attached to each sample for downstream analysis. Some strategies used to receive and track samples include the use of sample logs, in-house pipelines, and/or commercial information management systems. Sample tracking approaches can vary per lab and largely depend on the number of samples processed, sampling frequency, the facilities and personnel involved in the wastewater surveillance system, and the budget allotted for sample tracking and data management. Sample storage is another aspect of sample handling. Proper storage is critical to ensure sample preservation. Samples are usually transported to laboratory facilities on ice. Specifically for SARS-CoV-2, the CDC recommends storing samples at a temperature of 4 degrees Celsius immediately upon arrival and processing them within 24 hours to avoid degradation. Since human pathogens can be present at low concentrations in wastewater, concentrating large volumes of wastewater is vital to capturing a sufficient amount of the target in each sample. There are several common methods used to concentrate microbial targets in wastewater. The first method involves precipitation, namely precipitation with polyethylene glycol or PEG. PEG precipitation works by altering the solubility of the target, resulting in precipitation. In membrane absorption, the target is absorbed to a negatively or positively charged membrane filter by charge interaction and subsequent removal from the filter through chemical or mechanical processes such as elution or bead beating, respectively. Ultrafiltration is a pressure-based method which relies on the pore size of the membrane that the wastewater is being passed through. Depending on the pore size, the target will remain trapped on the membrane while water and other substances are allowed to pass through. And lastly, ultracentrifugation works by concentrating the microbial target down into a small pellet, usually through a series of centrifugation and resuspension steps. There are several things to consider when choosing a concentration method. These include cost, scalability, percent recovery, processing time, sample type and volume, the expected concentration of the target in the sample, wastewater composition, and the morphology of the target. Following concentration, samples undergo nucleic acid extraction to isolate DNA and RNA. Common extraction methods include organic extraction, where nucleic acid isolation is achieved by adding a series of chemicals to the sample to digest proteins and non-nucleic acid cellular components, and to separate out these components from the nucleic acid so that the nucleic acid can be easily removed and transferred to a separate tube. Magnetic bead extraction is a method that uses magnetic particles to bind to the nucleic acid and aid in isolation. And spin column techniques use centrifugation in a series of buffers and salts to promote binding of nucleic acid to the respective membrane. Nucleic acid is then eluted with an elution buffer or nuclease-free water. The extraction process can be either manual, usually with the use of commercial kits, or automated using commercial automated purification systems. Whether it is manual or automated can also depend on the method being used. There are several factors to consider when choosing an extraction method, including sample type, cost, throughput, compatibility with the sample type, and DNA and RNA yield. Lastly, purified nucleic acid can be quantified in wastewater typically using polymerase chain reaction techniques, or PCR. These methods can quantify specific targets of interest using a set of primers and probes. There are two main PCR methods used in wastewater surveillance. These are quantitative PCR, or qPCR, and droplet digital PCR, or ddPCR. qPCR is considered a gold standard approach for quantifying microbial targets in wastewater, with ddPCR being a new technology that is gaining much attention in WBE due to some advantages it has over qPCR. Specifically, the ability to quantify a target gene without the need for a standard curve, and its robustness towards inhibitors, which can be common in wastewater samples. Still, there are many cases where qPCR is preferred over ddPCR, and several factors should be taken into consideration when choosing a method, such as the application of the study, the presence of inhibitors, and expected levels of the target in the sample. Now that we've reviewed the basic steps involved in quantifying microbial targets in wastewater for wastewater-based epidemiology and methods that can be implemented for each step, I'm going to play a short video that reviews the methods used in the Houston system to quantify SARS-CoV-2 in wastewater. Untreated wastewater samples are collected by Houston Public Works and Houston Health Department personnel from wastewater treatment facilities, lift stations, and manholes around Houston, Texas, and are transported on ice to Rice University and the Houston Health Department laboratories for analysis. Samples are generally received in barcoded label bottles containing information about the type of sample, facility, and date of collection. Mixing of samples is important to ensure samples are homogenized before being aliquoted into their respective centrifuge tubes in preparation for centrifugation. Centrifugation is an effective method used to separate solids in a wastewater sample from the supernatant or liquid fraction and further purify and concentrate viral particles prior to concentration. Samples are stored at 4 degrees Celsius until further processing. Proper storage of samples is important to minimize viral degradation and increase the utility of wastewater surveillance. When ready to be concentrated, samples are scanned into a laboratory information management system, or LIMS, that allows us to easily and effectively track samples and attach associated data. Viruses are present at very dilute concentrations in wastewater. There are several different methods that effectively work to concentrate viruses and viral particles in wastewater samples. One method that our team and many other groups use is electronegative membrane filtration. Electronegative membrane filtration with flat or cartridge membrane filters can be used to concentrate viral particles from large volumes of wastewater. It works by binding the viral particles to the membrane filter, usually with the aid of a salt such as magnesium chloride. This method has been effectively used to concentrate SARS-CoV-2 in wastewater. While the supernatant is typically used to concentrate SARS-CoV-2, other viruses are known to adsorb more effectively to the solids fraction. With the addition of lysis buffer, solids can be resuspended for further processing. After filtration, electronegative membrane filters are transferred to sample tubes containing glass beads and lysis buffer. Bead beading is a critical step for us to ensure cell lysis and accurately quantify nucleic acid in wastewater. Centrifugation after bead beading is used to separate out the lyse cells and helps to prevent the transfer of glass beads and filter particles during extraction. Magnetic bead extraction uses small magnetic particles to capture nucleic acid from a lyse sample. An automated nucleic acid purification system is used to carry out extraction. There are several benefits to using an automated purification system, including a decrease in processing time and potential for contamination, and an increase in throughput and reproducibility. After the nucleic acids are extracted and purified, target genes are quantified in the sample. Our group uses digital PCR, and specifically droplet digital PCR. This method uses water-oil emulsion to form partitions that separate the nucleic acid molecules into individual droplets. SARS-CoV-2 is quantified in each wastewater sample using a one-step RT-ddPCR assay. Purified wastewater samples are transferred to PCR plates using an automated liquid handling workstation before being loaded into a droplet generator to form droplets. Each droplet functions the same as an individual test tube where the PCR reaction can take place. Bioconcentration data is then read on a droplet reader and analyzed using QuantiSoft software. The benefits of ddPCR are that it's a sensitive quantification method that is robust to inhibitors and provides an absolute count of target genes without the need for standard curves. In summary, different methods can be used to detect and quantify microbial targets in wastewater, and several factors should be considered when choosing a method, such as sample type, processing time, and target concentration in wastewater. Proper storage of samples is important to reduce degradation and increase the utility of wastewater surveillance. The use of a laboratory information management system or another system to effectively track samples can help improve workflow efficiency and organization. Electronegative membrane filtration can be used to concentrate viruses from large volumes of wastewater and has been used for SARS-CoV-2. There are several benefits to using an automated nucleic acid purification system, including a decrease in processing time and potential for contamination, and an increase in throughput and reproducibility. And finally, droplet digital PCR is a sensitive quantification method that is robust to inhibitors and provides an absolute count of target genes without the need for standard curves. If you're interested in learning more about sampling, analyzing, and integrating wastewater data into public health interventions, check out our other training videos on our website. Support for this program was provided through a grant by the CDC. The Houston Wastewater Epidemiology team would like to thank you for viewing this training video.